Today we'll be presenting on polycystic kidney disease. I'm Ryan.
I'm Nathaniel.
I'm Nathan.
And I'm Vikram.
So a brief overview of cell-to-cell communication. Basically, it coordinates cellular activities through the signal transduction pathway. Signaling molecules, also known as ligands, binds to the protein receptors, which are usually on the surface of the plasma membrane, and that's known as reception. Then transduction is how the cell responds to ligand binding, usually through protein phosphorylation catalase by kinases, enzymes that transfer a phosphate group from ATP to another molecule. And then this also usually causes a cascade of events. And what happens at the end of this cascade is the response.
So this is specific information. There are two proteins called PC1 and PC2, which act as G protein-coupled receptors and ion-gated channel, respectively. The PC1 use C-type lectin as a ligand, and PC2 gets a signal through a density-dependent factor. In PC1, the transduction stage leads to the kinase junct N-terminal and protein kinase C to produce C-amp and tubulogenesis as a response. In the PC2, the transduction stage CA2+ binds to the BRAF along with C-amp to regulate epithelial cell growth. So over here in this picture, you see PC1, which was the protein G protein coupled receptor, and over here is a membrane domain. So we have the GPS domain, the gain domain, and the PKD domain is what gets mutated. But over here, as I said before, this is the C-type lectin, which is the ligand. So PC1 can secrete this, and then it'll be attached to other PC1s, so it kind of regulates everything. And then over here, obviously, is the G protein, which in turn, through the cascade, produces tubulogenesis and C-amp. Then over here, we have PC2, which has the calcium ions that pass through, which again joins with the BRAF and the C-amp to regulate epithelial cell growth.
The inheritance of polycystic kidney disease depends on two genes, PTK1 and PTK2. The mutations of these genes cause two main types of polycystic kidney disease, autosomal dominant and autosomal recessive. When it comes to ADPKD, a single mutated PTK1 or PTK2 gene from a parent will cause each offspring to have a 50% chance of getting the disease. This occurs in 90% of recorded cases. However, in 10% of cases, children will develop the disease for a new mutation in their genes, and this only occurred in families without a history of the disorder. Only one parent has to have the mutated PTK1 or PTK2 gene in ADPKD. ARPKD, or autosomal recessive polycystic kidney disease, on the other hand, requires both parents to have mutated or abnormal genes to pass on this form of the disease. Two altered PKHD1 genes are required to become infected. In this case, the parents are only the carriers, and they are not affected by the disease, and the offsprings are, and each has a 25% chance of catching the disease if both parents carry them. On the first graph here, you can see that ADPKD is happening in a simplified version. One parent will have it, and on 4 children, the gene can spread and give to 2 of them, which makes it a 50% chance. On the second graph at the bottom, you can see ARBKD happening, where the unaffected parents are each carrying the gene, have 4 offsprings, and out of those 4, only one is affected, makes it a 25% chance.
There are two genes responsible for polycystic kidney disease, PKD1 on chromosome 16 and PKD2 on chromosome 4, which are responsible for producing the proteins polycystin-1 and polycystin-2. These two proteins work together to regulate the structure and epithelial cells in the kidney, essentially triggering the formation of kidney structures, allowing for the filtration of blood. In a cell with a mutated PKD1 or PK2 gene, it does not properly signal that this cell is a kidney cell. Either the PC2 is mutated and does not regulate the C-amp from overproducing epithelial cells because the calcium plus ions does not interact with the BRAF with C-amp, or the PC1 mutates, which does not produce the C-amp or the epithelial tubes as structures for the kidney. This causes the kidney to form a cyst full of waste. These cysts then interfere with other kidney cells, eventually causing failure of the kidney. Only one of the genes has to be mutated for polycystic kidney disease to actually form. While things may work with the other working copy of the gene for a time, as time progresses, the chance that a mutation will happen is only going to increase, virtually guaranteeing polycystic kidney disease formation.
So as the name suggests, polycystic kidney disease causes the formation of cysts in the kidney, but it can also cause cysts in other areas such as the pancreas, liver, and ovaries. The cysts in the kidney increase both the size of the kidney and the abdomen, usually causing back pain. The increase in kidney size results in a higher blood pressure and impairment of the kidney's ability to remove harmful toxins from the bloodstream. As Nathan mentioned, in the majority of cases, the kidneys will fail, and this can cause intracranial aneurysms and abdominal hernias. as well as heart disease in two main forms. One being the left ventricular hypertrophy, where the heart's left pumping chamber has thickened and can't pump blood as efficiently. And the other is mitral valve prolapse, where one or both of the mitral flaps are bulging, and this causes blood to leak backwards. And the other common symptoms include headaches and blood in urine. And this is the works cited. Thank you.